This video is for my biology for health science majors. In this video, we'll talk about lipids. Now, lipids are hydrocarbons, which means that they're made out of almost entirely carbon-hydrogen chains. Lipids contain twice as much energy as polysaccharides. So that makes them very important in long-term energy storage. We store most of our energy as fat, not as glycogen. If you were to store all of your energy as glycogen, you'd have to be twice as heavy. That's okay for a plant that is stationary, but if you have to be mobile, you want to be trim and light. So the more efficient energy storage is important in animals. Lipids are insoluble. They are hydrophobic, water-fearing compounds because they are nonpolar. They have no positive or negative electromagnetic charges to attract water. So lipids, like oils and fats, these are hydrophobic. They will not mix in water. And this is because water is attracted to water to the exclusion of hydrophobic molecules. So remember, here's water molecules. This is a single water molecule here. And these little arrows indicate that the electrons are always, spending a little more time around the oxygen. The oxygen's hogging those electrons. Electrons are negatively charged, so that gives the oxygen a slightly negative charge. And without the balancing forces of the electrons, the hydrogens are left with this partial positive charge. So that causes hydrogen bonding between the water molecules. The positive charges on the hydrogen of one water molecule will be attracted to the negative charges on the oxygen of the neighboring water molecules because opposites attract. So water molecules are cohesive. They're attracted to each other. And they are attracted to each other to the exclusion of oil. So here's oil on the top of water. And if you were to mix that up, you'll notice that, then you'd set the glass or the jar back down and the oil separates from the water. Why? It's not because water repels oil. There's no repulsion. It's just that water is attracted to water to the exclusion of oils. They're just being exclusive. So here's a water molecule lost. These are the oil molecules. So here's all the lipids. And here's a water molecule, the negatively charged oxygen and the two positively charged hydrogens. And here's the other water molecules down here. So any water molecule that's lost in the oil, it's going to be pulled out of the oil by the other water molecules who, by electromagnetic forces like magnets. It's like each of these little water molecules are magnets that attract all the other water molecules, and they pull the missing the lost water molecule, they pull it out of the oil. So they're not repelling the oil. They're just all clustering together and excluding the oil because they're attracted to each other so much. Now, I know this discussion of the chemistry of water and oils seems hyper detailed, but it's actually really crucial for understanding how cells stay together, because the cell membrane that divides the outside from the inside of the cell is made up of oil. And the only reason it doesn't fall apart, even though the cell is mostly water, is because oils don't dissolve in water. They're hydrophobic. So understanding how the cell membrane works will rely upon your understanding of water chemistry and hydrophilicity and hydrophobicity. Now, lipids differ from carbohydrates, proteins, and nucleic acids in that they are not actually huge molecules, and they're not built out of monomers. They're not polymers. They're not made out of these repeating subunits. Lipids vary a great deal in their structure and in their function. We're going to be talking about three different types of lipids. We have fats, phospholipids, and steroids. Now, a fat is a large lipid made from two kinds of smaller molecules, a glycerol attached to a fatty acid. A fatty acid can link to a glycerol through dehydration reactions, and a fat contains one glycerol linked to three fatty acids. Now, glycerol is a 3-carbon sugar, so it's not a hydrocarbon. It is carbon-based, but it's a sugar, not a fat, not a lipid. The fatty acid tails are the long hydrocarbon chains. So a triglyceride has 3 hydrocarbon chains attached to a 3-carbon sugar. So that's why they're called triglyceride, tri meaning 3. So here is a diagram of the dehydration reaction. So you have the glycerol at the top here. That's that 3-carbon sugar. That's going to be C3H6O3. And then this is the fatty acid tail. And there is a hydroxyl group at one end, but the bulk of the molecule is hydrophobic. In fact, these are so long that despite this one little hydroxyl group up here, this chain is so long that it really doesn't dissolve in water. The hydroxyl group is just like a drop in the bucket. It doesn't have a very strong effect on how well this dissolves in water. So this hydroxyl group and this hydroxyl group form water, and the remaining elements, this carbon is going to attach to that oxygen there. And that happens 3 times to form a triglyceride. Now, the less saturated a fat is, the more liquid it is. And what do I mean by saturated? Well, if we go back to this diagram, these chains are saturated, meaning they are saturated with hydrogen. Carbon forms 4 bonds. So each carbon in this chain is attached to the carbon before it and the carbon behind it. And then if it's saturated, it'll have 2 hydrogens attached. So saturated fats are holding as many hydrogens as possible. This tail is not saturated because of this double bond. Because of this double bond between carbons, there's not 2 hydrogens attached to each of these carbons. There's only 1, because this carbon is bound to, there's bond number 1 to a carbon, bond number 2 to a carbon, bond number 3 to a carbon, and only 1 bond to a hydrogen. So that is unsaturated because it's not carrying as many hydrogens as it could. The more saturated a fat is, the more solid it is. The more unsaturated it is, the more liquid it is, because of how they stack. So if you think about papers, like printer paper, when you get a new pack of printer paper, all the papers are stacked neatly on top of each other, and they hold their shape really well. That's like butter. That's like solid. But if you were to take one of those papers and crumple it up and then toss it down on the floor, and then crumple the next one up and toss it on top of it, crumple the next one up and toss it on top of it, and just do that to the whole ream of printer paper, they're not going to stack so nicely. Those crumpled pieces of paper are going to tumble over each other. That's what makes a liquid. So these unsaturated fats, they don't stack together very well. They're always going to be falling over each other, and that is what makes them liquid. So olive oil is highly unsaturated and butter is highly saturated. Saturated fats are bad for you because they can solidify even at body temperature, meaning they can solidify inside your blood vessels, and that can clog your blood vessels. Trans fats are even worse than saturated fats. Trans fats has the hydrogen has this double bond like an unsaturated fat, but the orientation of these hydrogen bonds is opposite each other. So if unsaturated fats are better for your health than saturated fats, it seems like trans fats should be okay too because they're unsaturated. Why should they be worse than saturated fats? And the answer is, again, because of the molecule's geometry. It all just comes down to geometry. And how these molecules physically interact with each other. So here's a saturated fat. They could stack on top of each other like a nice stack of paper. Here's an unsaturated fat, which is like a crumpled piece of paper, especially if you have more than one of these double bonds. And here's a trans fat. These, because of their shape, they lock together. So if I were to draw how they stack, so here in the saturated fat, they stack like a stack of printer paper. Unsaturated fats can't stack. They're all like this, making weird crumpled shapes with each other, and they don't stack at all. That's why they're liquid. These trans fats will lock together. These ridges will just lock into place. They will be even more solid than the saturated fats. So trans fats are worse than saturated fats. And they are produced by trying to artificially saturate fats in a laboratory. Phospholipids is the second type of lipid that we are going to talk about today. These are what build cell membranes. This is what builds the cell barrier between the inside and the outside of the cell. They are structurally similar to fats. They contain a glycerol, that 3-carbon sugar, but they only contain 2 of those fatty acid tails. And then on the 3rd carbon of the glycerol, you have a phosphate group, which was one of those functional groups I told you about. So here's a picture of a phospholipid. So here's this phosphate group. And because of its negative charge, it is very hydrophilic. Then you have these long fatty acid tails. There's two of them on a phospholipid, and those are hydrophobic. So these molecules are what we call amphipathic. They have a hydrophilic part and a hydrophobic part. That means an amphipathic molecule is one that has a region on the molecule that's hydrophilic, that dissolves in water, and another region on the molecule that's hydrophobic, that separates from water. And so what happens in the cell membrane, and we'll talk about this more in a future chapter, but the hydrophilic heads are attracted to water, so they face the water on the outside of the cell and on the inside of the cell, while all the hydrophobic tails cluster together. So phospholipids cluster into what we call a bilayer, bi meaning two, a bilayer of phospholipids. The hydrophilic heads are in contact with the water of the environment and the water on the inside of the cell. And then the hydrophobic tails cluster together in the center of the bilayer away from water. So there is water inside and outside the cell. The charged hydrophilic heads are attracted to the water on the inside of the cell and on the outside of the cell. And the oily, fatty tails are going to cluster together, facing each other, oriented away from water. And that creates a barrier between the inside and the outside of the cell. So bilayers like this, like the ones that form cell membranes, they actually form spontaneously when you mix amphipathic molecules like phospholipids in water. You can just take some phospholipids that are not part of a cell, just some phospholipids by themselves, shake them up in water, put them on a microscope slide. They will spontaneously form bilayers like cell membranes. Now, those are not making cells because a cell has to have DNA and stuff like that on the inside. But I'm just saying that phospholipids will form a membranous structure like this just due to their chemistry alone. Doesn't require any intervention from a living thing to form a structure like this when phospholipids are mixed with water. Finally, we get to the third type of lipid I wanted to talk about, which is steroids. So steroids are a type of hormone. You have stress hormones like cortisol. Those are steroid hormones. And then you also have sex hormones like estrogen, progesterone, testosterone. Those are all steroid hormones. Now, cholesterol is a really important component of the cell membrane. We just looked at the cell membrane made out of phospholipids. Cholesterol is also in there, and that modifies how fluid or how solid the cell membrane is. And cholesterols are also the starting material, the precursor for steroids, including sex hormones. So this is cholesterol, the precursor to steroids, and it's also a lipid. And remember, these corners are all carbons attached to hydrogens, right? So the carbon, these are all carbon rings where there's no other element indicated, assume it's carbon. And here I bring back these sex hormones, testosterone and estradiol. These are lipids because they are hydrocarbons. There are a couple other elements that are not hydrocarbon, these functional groups attached, but the bulk of this molecule is just a hydrocarbon, which makes it a lipid.